Okay.
This is Baron Curtin and Heather Geiger, and we'll be presenting our Project 2 for Data 620. So just an introduction, this time we were asked to work with a two-mode network data set. And one example of that is gene sets, where you have normally if you work with gene expression data, you'll have this whole list of genes, and you can categorize them according to their functions. So if you have a whole big list of genes, you can kind of narrow it down. And we'll be using the molecular function gene sets here. And you'll have a category such as cyclic nucleotide phosphodiesterase activity, and then whatever genes are involved with that. And so we're going to analyze this as a network to see which gene sets have, relatively speaking, more genes in common than you would expect by chance. And Baron, you want to do this part?
Sure. So first things first is that we have to read in the data. The way the file format was structured is that it had to be split into two parts. There were like numerous columns. The first one was part of the gene, and then the second column we did not need. Third column we did. So we read all of that in. We built a generic network X graph and added all of the all the nodes from the genes keys that we in a dictionary that we created. Again, we're just adding all of the nodes from the gene set database. We added all the edges, and that was done from converting a list of tuples to a dictionary. Drew the graph. The graph is rather large, but it might also be large because of just the names of some of these genes are just really long. Can't really discern too much from the graph just because there isn't much coloring done.
And so then the next step, since this is a two-mode network, you need to do projection to make it into a standard kind of where you can analyze it the same as a one-mode network. And if you do a weighted projection onto the gene sets, then the edge between any two gene sets will be the number of genes that they have in common. The only thing is, so if you have a lot of genes in common between two gene sets, if they're very large, that's not necessarily super meaningful. Because just by chance, if you chose any random 1,000 genes from your whole annotation, you're going to find a lot in common. So we got those weighted edges. But then we also wanted to normalize by the size of the gene sets. And normally it's actually a statistical test where you would say if you took two samples of those sizes by chance, you could get a p-value. We had too many edges here where we couldn't apply that test in a reasonable runtime, but we did normalize for any given edge between two gene sets, we normalize by the mean of the number of genes in each of those two gene sets. Now, Baron's going to talk a little bit about applying the island method to reduce the size of the network here.
Yeah, so first things first is that we had to get the weights of all of our genes into a dictionary, and we had to then set those attributes to the edges in our graph. So the next part discusses a little bit on parameter tuning. So part of the island method requires you to tune some parameters as far as the weight that you want to trim. So we decided to use mean, or see what the mean and the median told us about the mean weights and median weights. And those are two of the parameters that we had in consideration. From the mean and the median, you can see that the the weights are skewed right. But we ended up going with the mean weight just to trim the graph a little bit further. So then we set up these functions actually come directly from the textbook, but they've been modified to use some of the data that is unique to this specific data set. So weight norm is a thing that a feature that we extracted out of this data set from finding the normalized weights of the data set of each edge. So that's unique to the function here, but more or less it's strictly from the book. Also, the other alteration we had to make to the function is in the actual island method function, which uses NPA range, because when we normalized the weights, they became floats, and the regular range function from Python does not allow the use of floats. So we're utilizing that numpy function there. Afterwards, we get all of the connected components of our subgraphs, use our island method function to get all of the different islands. And then we just wanted to loop through all of the islands, print out the pertinent information, which included the threshold that we filtered for the islands, the size of the graph, and then the connected components of each graph. Do you want to discuss this part?
Sure. Yeah, so you see as you increase the threshold, it reduces the number of gene sets that are left based on the normalized edge weights. One thing, in the textbook, they used 5 iterations, but we still had over 300 gene sets left after the 5 iterations, so try just increasing the iterations further. And if you go to 10 iterations, then by the end, you can go down to 81 gene sets, which is a lot more manageable. And so by the end, if you do 10 iterations, you have 81 gene sets and 38 islands. And if you look at the gene sets that are left after that filtering, you can see there's a mix of different gene set sizes left. So you have a few of those really big gene sets that are left after filtering based on the normalized edge weights, meaning that they have, even normalizing for how big they have a lot of genes in common with other gene sets. But you also have some smaller gene sets left. One thing is that, so after the filtering by the normalized edge weights, a lot of these are more general molecular functions. So for example, cell-cell adhesion, regardless of what type of tissue or cell you're working with, it's going to have to stick to other cells. So it makes sense that the genes involved in that might also be involved in other more specialized functions. But you're going to have these more general type genes in those functions, so it makes sense that they would also be found in other gene sets. A few of these are just the quirks of the annotation. For example, these 3 categories, you see this icosanoid binding has 6 genes, and then you see 5 and 6 for these other category names. These are actually almost the exact same molecular function. 5 genes are the same, found in all 3 of these categories, highlighted on the screen here. So that actually, in terms of a network analysis, here we looked for the ones that were, we wanted to look for the highest normalized edge weights, but actually if we were trying to do this for bioinformatics, we might want the opposite. We kind of want to maybe remove these gene sets, or at least take only 1 out of the 3 of them, since it's kind of redundant information. But for network analysis, these would be the kind of gene sets we want to keep because they have a lot of connections to other gene sets. And that's about it.